Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. The paper that we looked at yesterday by Kamal offered up an extremely intriguing idea that APP potentially could be a trailer hitch that links kinesin to its cargo. If that's true, that would be extremely exciting. First of all, it assigns a function to amyloid precursor protein, and people have been trying to figure out what that molecule does for decades now. And second of all, it implicates kinesin in Alzheimer's disease. And it suggests that if you could target the transport process or the motor protein itself, that maybe you could have an effect on the disease. The group that wrote the paper that we're going to look at today, Lazarov and colleagues, were very intrigued by this idea, so much so that they just decided to repeat the experiment, because if it was really true that kinesin links to APP, that would be a fantastic finding. So the title of the paper is Axonal Transport, Amyloid Precursor Protein, Kinesin 1 in the Processing Apparatus, Revisited. And the important figure in this paper is Figure 2, Panel C. So let's take a look. To start out, I'd just like to reiterate the technique that they used in both this study, Lazarov, and in the study by Kamal. We said that they started out with mouse brain tissue. So mouse brain. And they ground it up. It's biochemistry. So they made their milkshake, and the scientific term that they use is a lysate. So they broke open the cells, they added some buffer, and homogenize the sample. And then, of course, when they spin it down, they end up with a pellet and a supernatant. The first test tube that was before the centrifugation is called the lysate. In this paper, they use a sample of lysate, so I wanted to make that clear. So then they can take the supernatant and put it in a new tube. So this whole tube is supernatant. And we said that that gives you soluble proteins and other components of the cell. And then to do the immunoprecipitation, they take an antibody against a protein that they want to look at, like amyloid precursor protein, and they bind it to a bead. So now you have antibodies bound to the bead, and they add those to this supernatant. And they would then bind, of course, amyloid precursor protein. Then when you spin these beads down, the bead would have the antibody, the APP attached, and then anything else that's attached, physically bound to APP. And in this study, they're specifically interested in determining whether kinesin heavy chain or light chain or both are associated with APP. So you'd have the light chain and then the heads of the kinesin and the tail connected to the light chain. Then they do a Western blot. And let me just erase what I have here. Okay. And I know you guys know this, but let's just go through it anyway. You have a protein gel, and you have different immunoprecipitation experiments. You pull one sample down with antibodies against APP, and then a sample down with a light chain, a kinesin light chain antibody, and then do an experiment where you pull down with a kinesin heavy chain antibody, and so on. And those samples are loaded into the well, and then electricity applied so that the proteins run down the gel. The smallest proteins run the fastest, and the larger proteins run the slowest, and then there's proteins in between of different sizes, of different molecular weights. And once that banding pattern is established, it needs to be transferred onto a piece of nitrocellulose, which you guys know is more or less paper. And you run the electricity through the gel, and that transfers the banding pattern onto the piece of nitrocellulose, and then you just throw away the gel. The part that I felt I wasn't totally clear on the other day was, now that you have your piece of nitrocellulose with your bands of protein on them, this gets incubated in a solution with the antibody added to it. And I didn't mention that it was in solution. I wanted to make that clear. So the antibody is just washing around in this solution, and it's looking for whatever antigen it was raised against, like amyloid precursor protein. And the primary binds to a specific site on the protein, and then a secondary antibody binds to the first, and the secondary antibody has something conjugated to it, and it could be radioactivity, it could be a fluorescent molecule, but for the sake of the conversation, they also make them that just turn color. It's colorimetric, so you end up with a nice purple band. And at any rate, if we were going to probe this for APP, you would get a nice band for APP. And then I just mentioned that for the sake of the figure and saving space, they just take a razor blade or a pair of scissors or something like that, and they cut out the region where they know the APP is, because they know the molecular weight of APP, and they run a known standard of different sized amino acids, so they can figure out that this band is APP. Plus, it's being recognized by the APP antibody, which is another piece of evidence. But they know that this part doesn't have anything on it, or this part here, so they just can throw that away. And then they take this strip and they make the strip part of the figure, like this. Okay, so I'm going to erase this, and let's go on and talk about the figure itself. Of course, this figure is very similar to the one published in the Kamal paper, because they're trying to just simply repeat the experiment. And across the top, they write the sample or the antibody that was used in the pull down. So the first lane actually is labeled L. Whoops. Is labeled L for lysate. And the lysate, again, is just a sample of the homogenate. It's total mouse brain ground up in buffer. So this serves as a positive control, because every protein in the mouse brain is present in the lysate, so that should include kinesin, amyloid precursor protein, and the kinesin light chain. The next lane is a sample of just the beads, that is, the beads used in the pull-down experiment, and there's nothing attached, they're just naked beads. So that's a negative control, because you wouldn't expect anything to get pulled down because there's no antibody used in the experiment. Then they use normal mouse IgG, which is mouse antibody, of course, but it wasn't, this particular IgG is not directed towards any of the proteins that we're studying in this particular case. Then there are a number of kinesin heavy chain antibodies, and the first one is called SUC4, and I'm pretty sure that was against a Searchin kinesin. But at any rate, this is kinesin heavy chain. The other kinesin heavy chain antibody is called H2. So this is kinesin heavy chain. And then they have a variety of kinesin light chain antibodies. And the first one that they have is the 6390, which was used in the Kamal paper. So this is kinesin light chain. And then they have one called L1 and L2. And those were antibodies specific for the two different versions of the light chain that was studied in the Kamal paper. So this is kinesin light chain antibody, and this is a specific kinesin light chain antibody. And then they have a lane that's called all. So they just added all three of these antibodies together and made a lane that has all of those light chain antibodies. And then they have a series of APP antibodies. So APP, and the first one is called 22C11. One is C-terminal APP, and the last one is called JH. But at any rate, all of these are APP. So this is APP, and this is APP. So they have 3 APP antibodies, they have 3 light chain antibodies, and then they have a lane where they add them all together. They have 2 kinesin heavy chain antibodies, and they have a series of controls. So then after they do the pull down, they do a Western blot, of course. And they write on the side of the Western blot the antibody that was used for the Western. So these are the antibodies to do the pull down, and these are the antibodies for the Western blot. The first antibody that they use is the kinesin heavy chain antibody. And when they do the Western, you would expect kinesin heavy chain to be in the lysate, because the lysate is all of the proteins in the brain, in the mouse brain, so they get a nice band. Then you wouldn't expect beads alone to pull down the kinesin heavy chain, or anything else for that matter, so it should be blank, and indeed it is blank. That's a negative control. The other negative control is the mouse IgG that's not raised against any of these proteins that we're interested in, and again, you don't see a protein there. But if you pull down kinesin heavy chain with SUC4, then you would expect to see the heavy chain in the Western blot, and they do see that. Then, if you pull down with a different kinesin heavy chain antibody called H2, of course, you would also expect to see the heavy chain in the Western blot, and indeed they do. Now we have 3 light chain antibodies, 6390, L1, and L2, and if these light chains are all bound to the kinesin heavy chains, you would expect to see heavy chain in each of these 3 pull downs. Actually, these 4 pull downs, because we also have all. And that's what we see in the figure. There's the 6390, the L1, the L2, and all. Now, if APP is associated with either the kinesin heavy chain or the kinesin light chain, because we know that the heavy chain and light chain are bound together, then you would expect to see kinesin in the APP pull downs, if it's true that kinesin is bound to APP. But in this experiment, this revisiting of this idea, they do not see a band for kinesin heavy chain in the 3 different APP pull downs. And this contradicts the result of Kamal. Now if they do a Western blot looking at the light chain, with the 6390 antibody, which is a light chain antibody that we know recognizes both versions of the light chain. In the light lysate, you would expect to see both bands, which they do. In the beads, a negative control, you shouldn't see anything, which they don't. The IgG, mouse IgG bound to the beads, doesn't pull anything down. It's not directed against any of the proteins that we're studying. They're studying. Now, if they pull down with these 2 kinesin heavy chain antibodies, you would expect to see the two light chains. And they do, for this heavy chain and for this heavy chain. And then we know that the light chain 6390 recognizes both. So if it pulls down both and then you probe it with the same antibody, you should get both bands. And then it turns out that the antibodies to L1 and L2 were raised against each of the two specific kinesin light chains. So the light chain 1 only recognizes only pulls down one of the two light chains, and the one that it pulls down, of course, is L1, and the other light chain antibody pulls down L2. And so it turns out that that means that the two different forms of the light chain are not on the same kinesin heavy chain. Sometimes the kinesin heavy chain has the L1, and sometimes the kinesin heavy chains have the L2. So there's two different makeups in that situation. And then finally, if you put all of the light chains together, you would expect to see both bands, and they get both bands. But at any rate, the point here, if you look at this data, is that if you pull down with kinesin heavy chain or kinesin light chain, you get both the kinesin heavy chain and the kinesin light chain, because obviously the kinesin heavy chain is bound to the kinesin light chain. If you look for the light chain using the pull downs with 3 different APP antibodies, in no case was the light chain detected, indicating that the light chain is not bound to APP. They then do 2 more Western blots looking at APP. And the first is the C-terminal APP antibody, and the other one is called JH APP. And you would expect to see APP in the lysate, which they do, a very nice band. You would not expect anything here in the beads, they don't. The IgG, they don't see anything. And then if kinesin heavy chain were bound to APP, when you pull the kinesin heavy chain down, you should see APP here. And they don't see that. They don't see it for either of the 2 heavy chains, and they don't see it for any of the 4 columns that are looking at the light chains. They also, for some reason, don't see APP when it was pulled down with 22C11. So this antibody just simply didn't work. But when you pull down with a C-terminal APP antibody and you probe with the C-terminal antibody, you get a nice band for APP. And you also see a band when you pull down with the JH APP antibody. And they see the exact same pattern for the JH. They see it only in the lysate or only in these 2 pull downs that pull down APP. So there's nothing bound to APP. I mean, none of these kinesin heavy chains or light chains get pulled down when you pull down APP. Okay, well, what to say? Well, both groups can't be right. One has to be right and one has to be wrong. And the 2 things that stick out in my mind, first of all, is that in the Kamal paper, they didn't show the Western blot using the heavy chain antibody, as I mentioned yesterday. And they must have done the experiment, because that's what you're trying to determine, whether APP is associated with the motor, the part of the molecule that moves along microtubules. I mean, directly or indirectly, but that's the important point. And so they must have done the experiment, and the only reason that they wouldn't publish it is because it didn't come out the way they wanted it to, that it didn't show that for some reason, and it didn't make it into the paper. The second point is that this group, the Lazarov group, I think would be extremely happy if the Kamal paper were correct, because that group works on kinesin, or some of those individuals work on kinesin. In fact, Scott Brady was one of the early discoverers of the kinesin motor family. So he would be very happy if APP were bound to kinesin, either directly to the heavy chain or indirectly through the light chain. And so I don't think the group is necessarily happy in the end that they did all this work to repeat the experiment, and it turns out that they came up with the opposite result. So I tend to favor the Lazarov conclusion, even though I too wish that the Kamal paper were correct. And it's just how science works. You have to do your experiments, and once you have the results, you interpret them to the best of your ability. And I think that that's what Kamal and that group did, and then Lazarov came up with the opposite conclusion. So more work to be done. Okay, you guys, that's it for today. We have one more paper on Friday. I hope you're doing well, and I'll talk to you soon. Bye-bye.